The general principles of gene regulation in prokaryotes also apply to eukaryotes, but there are some differences. Eukaryotic genes are controlled individually and have more complex regulatory sequences than those of the lac repressor system. Look at the illustration. This region is called the Tata box. One of the most interesting features of a typical eukaryotic gene is the Tata box. The Tata box is a short region of DNA, about 25 or 30 base pairs before the start of a gene. This region contains the sequence TATATA or TATAAA. The Tata box binds a protein that helps position RNA polymerase by marking a point just before the beginning of the gene. Getting transcription started, which is shown in this illustration, is just one way that eukaryotic genes can be regulated. They can also be regulated by transcription repression. How are genes regulated in eukaryotic cells? By binding DNA sequences in the regulatory regions of eukaryotic genes. Transcription factors control the expression of those genes. Gene expression in eukaryotic cells can be regulated at a number of levels. One of the most critical is at the level of transcription by means of DNA-binding proteins known as transcription factors. Some transcription factors enhance transcription by opening up tightly packed chromatin. The chromatin is the material that makes up the chromosomes. So the DNA gets wrapped around these proteins and it's very tightly packed, which can make it difficult to transcribe. So these transcription factors will open it up and make it easier to read. Other transcription factors help attract RNA polymerase. Still others block access to certain genes, much like prokaryotic repressor proteins.